Our final speaker is Dr. Mustapha Sultane, representing G-Force in the Near East region. Dr. Sultane will discuss harmonizing import maximum residue limits for pesticides, highlighting regional challenges, the need for coordinated approaches and alignment with codex and international standards. Mr. Mustapha, the floor is yours.
Thank you, Dr. Ruba, for the introduction. Good afternoon, esteemed colleagues. It's a privilege to join you at the 4th Global Food Regulatory Science Symposium here in Dubai. This presentation explores a scientific and regulatory pathway towards harmonizing of import maximum residue limits, MRLs, for pesticide residues across the Arab region. The aim is clear: to protect consumers while facilitating fair and efficient trade. Today's talk, we will move through 3 key parts: the background and rationale for harmonization, a detailed case study, and a proposed framework and call for regional collaboration. Let us begin with a quick background why pesticide residues are such an important topic for regulatory science and international trade. Before that, I would like to thank Dr. Eli for making this presentation much, much easier. Pesticide residues are at the heart of food regulatory decision-making. They are more than laboratory outputs. They influence market access, consumer trust, and the public health policy. Differences in MRLs between countries, or even the absence of an MRL, can become unintentional barrier to trade. Sometimes, a pesticide used safely in one country may not be registered in another. When that happens, shipments can be detained or rejected even if the food poses no health risk. To bring scientific consistency to these decisions, the Codex Alimentarius Commission has set a clear guidelines. Codex relies on the work of the joint FAO-WHO meeting on pesticide residue, which is the JMBR, which is an expert body that assesses residues based on toxicological and agriculture data. GMBR evaluates both acute and chronic exposure compared to a health-based guidance value, such as the ADI, which is the acceptable daily intake, the amount that can be safely consumed over a lifetime, and acute reference dose, which is the amount safe to consume within a single day. These guidelines also accounts for results related to patterns of use, fate of residues, analytical methods, etc. JMBR also reviews these values periodically, ensuring they reflect the most current science and analytical methods. But what happens when there is no Codex MRL and no national standard in place, or even when a national standard MRL exists, but enforcing it may compromise the food security consideration? This is exactly where the concept of an import MRL becomes essential. Now imagine this scenario. A food shipment arrives at the border, a pesticide residue is detected, but there is no local MRL, and Codex has not established one either. This creates a regulatory vacuum. The product is detained, and the competent authority has no clear benchmark for action. In such cases, an import MRL, or import tolerance, provides a practical science-based solution. Before moving forward, it's important to define the term import MRL. As you can see on the screen, an import MRL is MRL set only for imported products, designed to meet trade and food security needs when neither national nor codex limits exist, but national food safety standards is achieved. This mechanism can also apply in cases where a food security concern arises even when an MRL exists, the product is of high economic value, or the pesticide is authorized abroad but not domestically registered. Import MRLs ensures that safety assessment remain consistent while allowing trade to continue without compromising consumer protection. To make such a decision consistently, a structured process or a decision tree is needed. The decision tree offers a transparent framework for evaluating import MRL requests. Let's walk through it step by step. As you can see, the first step is that the residue level non-compliant with national MRL or no codex MRL. So the first question we should ask is, is this level authorized in the exported country or by the exported national authority in this country? If no, then the answer is to reject the product. But if yes, we should request for the scientific justification for such registration or legal limits. So if justification is submitted, so we should review these scientific justification. Is it according to acceptable practices, transparency and risk analysis based? If no, then we reject the product. But if yes, then we should conduct a rapid risk assessment based on the national consumption patterns to confirm suitability, even if suitable or not. If the risk assessment conclusion shows a health risk, then we can reject the product. But if yes, then we accept the MRL as only import MRL, which is not designed for the domestic use, but only for the imported products. This approach would give us the opportunity, and this system would allow regulators to make defensible, evidence-based decisions while supporting trade continuity. With this framework in mind, the next step is to explore how a practical import MRL list can be established. A dedicated import MRL list would transform case-by-case decision-making into proactive, coordinated approach. Such list could be built by combining historical compliance data, systematic review of partners' practices, and industry-submitted justification supported by data from that decision tree. It's essential to clearly separate pesticide registration, which involves domestic use, from import MRL, which only applies to imported goods. This structured approach would allow competent authorities to review and update MRLs periodically using real trade and laboratory data. The result is a living system that ensures fairness, predictability, and transparency, attributes critical for both industry and regulators. Now, the foundation is clear. We can move to a real-world example, a case that illustrates exactly how this process works in practice. Our case study is about a practical case study about the scenario of tricyclazole residues in imported rice. And here we emphasize again on imported. We all know that rice is a significant contributor in our dietary habits in the Arab region, and any disturbance in the supply chain may consider as a food security issue. A shipment of rice was detained at the point of entry after laboratory analysis revealed that tricyclazole residues exceeded the national MRL. The detected levels was roughly 0.2 ppm. In this case, as we mentioned earlier, we can assume that no codex or national MRL or even the applicable MRL is 0.1 ppm, which means the consignment was technically non-compliant. However, all other pesticides tested in that shipment were well within the legal limits. Because of this single exceedance, the shipment was held under investigation pending a health risk assessment. Tricyclazole is a fungicide primarily used to control rice in rice blast disease, a major problem in paddy cultivation. It has been registered for agriculture use in several Asian and Latin American countries, including India and Brazil. However, it's not approved in the European Union, the United States, or Canada, primarily because of different regulatory philosophies and data requirements, not necessarily because of different toxicological evidence. As you can see, multiple jurisdictions have authorized the use of tricyclazole with an MRL 3.0 ppm, such as Japan, Brazil, New Zealand, and Australia. The assessment began with a simple question: was the expected health risk, what is the expected health risk from exposure to tricyclazole in rice at 0.2 ppm, and is this rice fit for consumption? To answer that, a standard risk assessment process was followed, starting by hazard characterization, and I won't repeat myself after what Dr. Eli has mentioned, and the second step is exposure assessment, and finally, risk characterization. This structured approach ensures that even when residues exceed the national MRL, regulators can determine whether the product actually poses a health risk to the public. We will start by reviewing the hazard value, then move to the exposure and risk characterization. EFSA have established toxicological reference values for tricyclazole. The acceptable daily intake, which is the ADI, is 0.05 milligram per kilogram body weight per day, representing the amount that can be consumed every day over a lifetime without appreciable risk. The acute reference dose is also 0.05 milligram per kilogram body weight, indicating the amount that can be safely ingested within a single day. These two numbers, both 0.05 milligram per kilogram body weight, are the scientific anchors for assessing any residue level of tricyclazole in food. In this assessment, those reference values provided the benchmark against which the estimated dietary exposure was compared. With those reference numbers set, let's estimate what an average consumer would actually be exposed to. As you can see on my screen, to quantify dietary exposure, three main data were applied from national consumption reports. And here, I want to emphasize on what my colleague also earlier mentioned about having reliable occurrence and consumption databases to boost the food risk assessment processes. The annual rice consumption equals 67.1 kilogram per person body per year, which translates to roughly 184 grams per day. The average adult body weight is 70 kilograms, and the raw to cooked conversion factor is 3, since residues are measured on raw grain, but consumptions data often refer to cooked rice. By calculating the daily intake of tricyclazole, which is the residue level, 0.2 ppm, multiplying by the daily consumption, which is 184 gram per day, the result is equal 0.368 milligram per day. Dividing this result with the average adult body weight, which is 70 kilogram, we get 0.0005 milligram per kilogram body weight per day. Expressed differently, the risk ratio represents 0.0105, which is nearly 1%. So let's translate those numbers into a clear conclusion for decision makers. At a residue level of roughly 0.2 ppm, the calculated dietary exposure represents only 1% of both the ADI and the acute reference dose. This means the exposure is approximately 95 times below the amount considered safe by international authorities. The conclusion, therefore, is ambiguous. The rice represents no appreciable public health risk under current consumption patterns. From a scientific standpoint, the product is safe for human consumption. The non-compliance arises solely from regulatory misalignment rather than toxicological concern. With the risk assessment showing no appreciable health risk, the next step was to define a proportionate, transparent risk management strategy. With the rice shipment was technically non-compliant, the scientific evidence indicated that the residue level posed negligible health risk to consumer. Given food security and supply chain consideration, 3 risk management options were proposed. First, to authorize a one-time release of the detained consignment based on the documented risk assessment results. This would prevent unnecessary food waste and disruption to the supply chain while keeping full traceability records. Second, to review the existing MRL for imported rice and consider alignment with internationally recognized standards such as New Zealand or Australia or any country that adopted the 3.0 ppm, a level supported by established good agriculture practices and risk assessments. Third, to develop a structured framework for evaluating and adopting import MRLs when justified by science. This framework would ensure that future decisions follows a predictable data-driven process. Together, these measures protect consumers, maintain public confidence, and minimize unnecessary trade disruption. Let's now look at what broader lessons this case study offers for our region. This example highlights an important distinction in regulatory science. First, regulatory non-compliance doesn't automatically equate to health risk. Second, this reinforces why scientific justification must guide MRL setting rather than relying purely on administrative or legacy limits. Third, a structured import tolerance or MRL mechanisms helps bridge these gaps. It allows authorities to evaluate whether residue detected, although not aligned with domestic MRLs, remain within a safe exposure range based on a toxicological data and dietary intake models and international standards. Ultimately, adopting a data-driven decision-making framework strengthens both consumer protection and market continuity, ensuring food safety without unnecessary trade disruption. Let's always remember that science-based policymaking is the bridge between safety and trade, not a choice between them. To sustain such science-driven system, institutional capacity and cooperation are essential. No single country can develop all the required data alone. Our region shares common consumption habits, agriculture practices, climate conditions, and even export and import behaviors, making cooperation both logical and efficient. By sharing exposure and consumption data, residue monitoring results, and analytical databases, countries can create a joint foundation for harmonized import MRLs. Such collaboration would not only support trade among Arab states, but also enhances our collective position in global codex discussions. Ultimately, these efforts lead to stronger, more resilient food safety system. In conclusion, harmonizing import maximum residue limit is not only a scientific necessity, but also a policy opportunity. It allows our region to ensure consumer protection while promoting predictable, fair trade. The process begins with detection, moves through structured assessment, leads to proportionate decision, and ends with continual review. Import MRLs serves as a bridge connecting science and regulations until full harmonization with Codex is achieved. By anchoring our decisions in shared science, transparency, and collaboration, we can build food systems that both safe and economically resilient. This is the vision to make the Arab region a model for evidence-based food regulation that protects its people and supports its producers. With that, I would like to thank you for your attention. Thank you so much.